Hello, welcome to today's LA Biotechnology Minireviews webinar series. Today we talk about vaccines. My name is Yvette van Eenenaan. I'm the CEO of Adelaide Biotechnology, the Health and Medical Innovation District in the heart of Adelaide, and I'm pleased to be the chair of the webinar today. So before introducing two fantastic speakers we welcome here today, I would like to acknowledge the Kaurna people, the original custodians of the land. And I host this webinar on this land today here, and I would like to pay my respect to the elders, both past, present, and future. I would also like to welcome our South Australian, our national and international attendants today. These weekly short webinars are really organized for researchers, for clinicians, students, entrepreneurs, and experts in industry that are here residing in South Australia or any other place across the globe. It's really to get a deeper insight in what's happening here in South Australia and help you link up with potential new partners, opportunity, and new knowledge. So the webinars will be recorded today as well, and we will place them on our website, our Adelaide Biotechnology website, so you always can go back to those recordings. But we, over the last 2 years, also recreated a kind of a TEDx-based database with all webinars around a wide variety of expert areas in the health and medical sector. So feel free to go there. I think we're over 100 webinars now at this moment to search for relevant areas you're interested in. So let's now zoom in on the topic of today. But before introducing two presenters, I'd like you to make you aware of the Q&A at the bottom of your page. And I'd like you to encourage to ask questions to the presenters today. I, as a facilitator, will read them and will propose them back to the speakers at the end of their presentation. So please feel encouraged to drop any question you have during or after the presentation. So now we go to our first presenter. Welcome, Professor Tim Hirst. Tim is an entrepreneurial academic with a very wide range of expertise in establishing new ventures, back companies. He's also the chairman and the CEO of GPN Vaccines. He's the executive chair of GPN Vaccines and has a very, very long list of director roles he has fulfilled successfully in the last decades. He's been the CEO of 30 million pre-seed investment funds, ANU Connect Ventures. And besides this impressive resume in leading company, he also held senior positions in universities in the United States, or United Kingdom, sorry, not the United States, the United Kingdom and Australia. He was deputy director of the University of Sydney. And to end this short summary of his resume, this year he was awarded Australian Angel Investor of the Year. So congratulations, Tim. Thank you for joining us here today. I will now hand over to you to talk about the need for pneumococcal vaccines.
Thank you, Yvette. It's a pleasure to welcome everyone to this webinar. And what I'm going to do is put up my screen and show you some slides on what we're actually doing. So I'd like to introduce GPN Vaccines to you. We're now a clinical stage company attempting to solve the problems of current pneumococcal vaccines. And what I'm going to do is introduce you to a new vaccine, which we call Gamma PN. Now, Streptococcus pneumoniae, which causes pneumococcal disease, is the world's foremost bacterial pathogen. And you see a picture of it there. It's a typical diplococcus in structure. It's got a cell wall and around the surface is a polysaccharide capsule. And it's a commensal organism that grows in the nasopharynx of many of us, and up to 90% of people are asymptomatically colonized by this bacteria in their nasopharynx. But it can invade deeper tissues, and when it does so, it can cause serious disease. For example, it can cause pneumonia. It can also enter the bloodstream and cause sepsis, and migrate to the brain and cause meningitis. And in children in particular, it's responsible for causing middle ear infections or otitis media. and up to 2 million people die from pneumococcal disease each year. And 20% of all deaths in children under 5 years of age in developing countries are caused by Streptococcus pneumoniae. And otitis media is extremely high in children. People estimate it costs as much as $5 billion per annum in the US as a consequence of lost productivity. And it's one of the reasons for the overprescription of antibiotics. Now, the capsular polysaccharide, which is on the surface of the bacteria, changes, and there are many different strains, and these are called serotypes. And there are 100 different serotypes out there. And the slide next to the picture of the pneumococcus shows the incidence of infection in the United States in adults caused by different serotypes. And the dominant serotype is serotype 3. And this is despite the fact that this serotype is supposedly being covered by a particular vaccine produced by Pfizer, which is called Prevnar 13, despite it being developed to prevent serotype 3 infection. There are very high incidence, and that's shown in the blue column. And Pneumovax 23 is another vaccine produced by Merck, and you can see that there are a number of serotypes that are causing infection despite there being a vaccine. And then there are non-vaccine serotypes that are not covered by any vaccine, and those are shown in the red bars. And so the third, fourth, and fifth most prevalent cause of pneumococcal disease in adults in the US is caused by serotypes not covered by the existing vaccines that are on the market. And these are those vaccines that are on the market. You can see Pneumovax 23 comprises the capsular polysaccharide from 23 different strains. So it's quite a complex vaccine to make. You have to purify the polysaccharide capsule and formulate it into a mixture of different capsular polysaccharides. But because carbohydrates elicit a T-cell independent response, it's a very poor vaccine. Pfizer, well, I should say Wyeth, before Pfizer bought Wyeth, developed a polysaccharide conjugate vaccine in which they conjugate chemically the purified polysaccharide to a protein carrier. In this case, it's CRIM197 or diphtheria toxoid. And that's a very complex process. So each individual serotype has to be purified, it has to be conjugated, and then all those individual vaccines have to be brought together and formulated into a final product. So it's an extremely complex vaccine. Vaxneuvance is a new vaccine from Merck, a polysaccharide conjugate vaccine, where they've added an extra two serotypes to the Prevnar 13 vaccine. And Prevnar 20 has recently been launched by Pfizer in an attempt to solve this problem of some of the serotypes not being effectively protected against. And there are new vaccines in development from companies like Vaxcyte and Affinifax, which are 24-valent vaccines. And there's a company called InventPrice, which has been supported by the Gates Foundation, which is developing a 25-valent vaccine. And that effectively encompasses all of these different serotypes here that are shown on the screen, plus one extra serotype. Despite this, even when that 25-valent vaccine is developed, it still won't provide protection against the third, fourth, and fifth most prevalent cause of disease, and probably won't be effective against serotype 3. So even when these vaccines have been launched, they're inadequate on the day that they're launched. And so a different solution is required. Now, this just shows you one of the other problems with the Streptococcus is that when you launch a vaccine, very quickly new serotypes emerge, and that's often because you displace the bacteria that are in the nasopharynx and create a niche whereby other serotypes can come in and take over. And this serotype 24F, for example, has now become the dominant serotype in Japan that causing disease in children. So it's a serious problem of very complex vaccines, inadequate serotype coverage, and even when you do launch a vaccine, you end up with a problem of different serotypes beginning to take over. And the one problem that I've highlighted already, but just illustrates the complexity of making these polysaccharide conjugate vaccines is shown here. This is from Pfizer's annual report in 2016. And what they said there was that the manufacture of Prevnar 13 involves 580 manufacturing steps, over 1,700 employees, 678 quality tests, 400 different raw materials, and more than 2.5 years to manufacture from start to finish. Prevnar 20 is even more complex. The average list price for a single dose of Prevnar 20 in the US is $327.39 and is really much too expensive for providing a vaccine in the developing world where the incidence of pneumococcal disease is very high. So a different solution is needed. And this just summarizes some of these take-home lessons. Pneumovax 23 is a poor vaccine. Serotype 3 is not covered by existing vaccines and continues to be a problem. Serotype replacement is occurring all the time. New polysaccharide conjugate vaccines have been launched, but they're not effective against some of the prevalent serotypes, cost effectiveness issues, and the newest pneumococcal vaccines are too expensive. So that just summarizes the situation, and there's a clear unmet need for a cost-effective pneumococcal vaccine. And this has been our approach, where we've actually dispensed with the focus on the capsule of polysaccharide and decided to look at the whole cell proteins as a basis of providing immunity. There was a very interesting paper published by Jerry Brown's group at UCL in London, in which they looked at what is present in gamma globulin. Gamma globulin is a pooled human serum, which is given to individuals that have a deficiency in IgG production. And these particular individuals are very susceptible to respiratory infections. And they asked the question, what is it that's in this gamma globulin that provides protection against respiratory infections, in particular against Streptococcus pneumoniae? And they came to the conclusion that it was antibodies in the serum directed against proteins, not against polysaccharide capsules, that were providing protection. This completely broke the dogma as to the basis on which you should develop a pneumococcal vaccine. And in fact, we'd already begun working on a whole cell vaccine, which elicits antibodies to proteins. And this is our approach. We take the whole cell, it's not got any capsule. We've engineered it with a number of mutations. It's very safe and easy to manufacture. And then we kill the bacteria by exposing it to high energy photons or gamma rays. And what that does is it inactivates the bacteria, it can't grow, but all of the proteins are maintained in their natural native conformation. And it's very easy to manufacture. So there are just 7 steps in producing this product. We grow the bacteria in a fermenter, we do some downstream processing, we vial it, we expose it to irradiation, and we produce the vaccine. The contrast in the complexity of this, or the lack of complexity, with the polysaccharide conjugate vaccines is quite striking. And this shows what happens if you vaccinate, in this case, rabbits, with 2 versions of the vaccine that we've been working on, called gamma PN2 and gamma PN3. Gamma PN3 is the one we're taking into the clinic. You can see that it produces a very robust IgG response. And then when you probe that serum in a sort of multiplex microarray system, you can see that the antibodies are binding to many, many different proteins that are present in the pneumococcus. So this whole cell vaccine elicits a very broad spectrum immune response to the pneumococcus. And if you take capsulated strains of the bacteria and add the serum and look to see, do these antibodies bind to encapsulated bacteria? The answer is yes, very high levels of binding. So these antibodies can find the proteins despite the fact there's a capsule on the surface of the bacteria. Now, one of the most important abilities of pneumococcal vaccines is the capacity to induce antibodies that are opsonophagocytic or opsonic antibodies. And these antibodies bind to the bacteria, and then together with complement and neutrophils, they're engulfed, and the bacteria are killed. And this has become, if you like, a bactericidal assay, in which has become a surrogate, if you like, endpoint, which points to the effectiveness of the vaccine. And in fact, regulatory agencies such as the FDA are now using the OPA assay as a basis for regulatory approval of expanded coverage PCVs, polysaccharide conjugate vaccines. And so what we've been doing is looking to see whether our wholesale vaccine will induce an OPA response. And the answer is it does. Very robust response, in some cases of much greater magnitude than the vaccines such as Prevnar 13 or Pneumovax 23. So we're getting very, very, very high opsonic antibodies produced by this. And that's really a unique finding, because other people have tried to do this previously and have not been able to achieve it. So we feel very confident that this product will be able to achieve the requisite type of immune response when given to humans to provide protection against pneumococcal disease. Now you'll see here that sometimes the assay doesn't work terribly well, and we've had some difficulties with this, which could be due to the batch of complement. And so we tried another method to look to see whether the antibodies that were generated in these animals would provide protection against a lethal challenge with serotype 3 Streptococcus pneumoniae. And this is shown here. So we've basically mixed the bacteria with the antibodies, and then we've injected those bacteria, 100 bacteria, into the peritoneum of a mouse. And what you can see here is that very rapidly, within 48 hours, the animals which have the bacteria which have been incubated with the pre-bleed pre-immune serum are all die within 48 hours, whereas those that have been treated with the gamma PN serum, 100% survival occurs. So we feel very confident that this vaccine will provide protection against serotype 3. So one of the other things that we've obviously been doing is focusing on manufacturing, and this has been done at Biocina in Adelaide. Much of our work is done in Adelaide, and Biocina have done a fantastic job in developing an optimized manufacturing process for us, including scaling up and producing a clinical grade batch. We've carried out toxicology on this material that's been produced in Adelaide at Charles River Laboratories in Edinburgh, and that went through fine. There were no issues of concern. We've had approval from Belbury to conduct a first-in-human clinical trial in Adelaide at CMax, which will take place in January. This will be a sort of fairly large 117 cohort randomized placebo-controlled double-blind study to look really at the dose that's appropriate for use to achieve safety, tolerability, and immunogenicity. And we're targeting adults 50 to 69 years of age. And this is the clinical trial design. There'll be samples taken over a 2-month period, both serum and PBMCs, to look at the cellular immune response as well as the antibody response. So in conclusion, this is the first vaccine that targets non-capsular antigens that elicits a high titer functional OPA response and is able to achieve that response against both current serotypes. Gamma pickle vaccine offering protection against all serotypes, including any new serotypes that may emerge in the future. And we hope to announce the headline results of our clinical trial from the work being done in Adelaide at the beginning of the calendar year 2024. Got a great board of directors, including Ishmael Kohler and Madeline Cox from the United States, James Payton, who is a professor at the University of Adelaide and Chief Scientific Officer of the company. Lauren Cox, some of you may know, is our Chief Operating Officer. We've got a management team again, Paul Rowland, based in Adelaide, Eve Singleton, who's a senior research scientist, and Elsa Rishi, who's providing input on the immunology side. And we've got numerous partners, including grant from the South Australian government. So we're based at the University of Adelaide. We work closely with ANSTO for the irradiation purposes. CMax is providing the phase 1 clinical trial unit. Manufacturing has been done at Biocena. Southern Star Research in Sydney is that we're working with. And with that, I'll say thank you and answer any questions.
Yeah, here I am. Thank you, Tim. Thank you, Fink. And also sharing at the end the broad range of partnership you're working and partnering with across our ecosystem. To finish your presentation, I have a question, and I encourage everybody to ask a question through the Q&A. It's at the bottom of your screen. It's Q&A. Just type your question, and I will look at it and hand it over to Tim. But one question related to the mRNA technology, everybody's very excited about, as you know. Could this be an alternative approach for creating a universal meningococcal vaccine?
Not at all. It won't work for a number of reasons. Obviously, the mRNA technology is designed to produce a protein. And if one were to go down the sort of targeting the capsule of polysaccharide, obviously you can't make a polysaccharide from expressing the mRNA. It's just you make proteins with mRNA. So you wouldn't get an anti-polysaccharide response, you would get an anti-protein response, but we think you need a response to maybe 50 or more different proteins, and it would be impossible to think of a mRNA vaccine where you had 50 different RNAs all needing to be formulated in order to get a broad spectrum response. We think that's going to be critical. In addition, this OPA response, which is so vitally important to the efficacy of the vaccine, appears to rely on some of the features which are intrinsic to the whole cell, because bacteria have pattern recognition features which activate the immune system in particular ways. And we think the feature of the immune response, which gives you this OPA adjuvant, commonly used in human vaccines, to boost the IgG response. We got a boosted IgG response, but we completely lost the OPA response. So that was really intriguing. And you would not be able to achieve that same OPA response with proteins generated by the mRNA technology. So we think we've got an edge on that. We don't think there's any possibility of the mRNA technology finding an application in pneumococcal vaccines.
Thank you for elaborating on that. I think some people have some more Q&A questions, but we're running out of time. So I would like to ask people joining us here today or looking online to contact Tim directly if they want to go deeper in some sort of partnership or discussions in the future. Thank you, Tim, for joining us here today. Then I will now go to our second presenter, Leanne Hobbs. She has over 25 years of experience in the consumer health care and the pharmaceutical and vaccine industry, both locally here and internationally. She is an Adelaide University alumni and returned to Adelaide in 2019, I believe. But before that, she spent many time in London, Melbourne, Belgium, working for GSK. And in her last role there, she was a global general manager for the HPV vaccine franchise. And she had oversight of many functions like R&D, medical, commercial, government affairs, finance, business development, supply chain manufacturing, very comprehensive oversight. And prior to that, she was dedicated in the roles as drug licensing and regulatory affairs, health economics, new product planning, sales and marketing. So thank you, Leanne, for joining us today. I know you took also a sabbatical during your career, and you spent some time in Switzerland, I believe. You volunteered as a mentor and pitch judge in the Mass Challenge, the startup accelerator in Switzerland. And that's where you really build up your knowledge of the investor and funding landscape for startup companies. We're very keen to see you back land here in Adelaide and have the role as CEO of Sementis. And please share with us more information about your companies, but also keen to hear more about Australia's most advanced viral vaccine platform you'd like to share with us today. I hand over to you.
Thanks a lot, Yvette. I'll just seek to share my screen. Okay, so hopefully everyone can see that. So thanks for the introduction, and thanks everyone for joining today. So I'll just jump straight into it. So for those of you who don't know us yet, you know, we're Sementis. We are an Australian public unlisted biotech company based here in Adelaide. The company was founded in 2009 by Dr. Paul Howley, who actually was ex-Bavarian Nordic and had a long history in the development of vaccinia-based viral vector technology, which explains a little bit the story of this presentation. We are a privately funded company predominantly with some very significant and loyal shareholders, but we also have received significant government funding and grant support over the last few years from the Medical Research Futures Fund and HMRC, more recently the DMTC and also CSIRO, so we gratefully acknowledge that funding. We are a preclinical stage company with a great ambition to be in clinic within the next 2 years. The current leadership team for Sementis is myself as Chief Executive Officer, as Yvette introduced. We also have our Chief Scientific Officer, who is Professor John Habel. He is the head of the Experimental Therapeutics Laboratory at the University of SA, but also as our CSO. And we have our Chief Operations Officer, who is Dr. Anne Collins, who's known to many of you in Adelaide, and no doubt nationally and internationally as well. She's ex-Trajan Main, has an amazing biomanufacturing background and just general multinational industrial experience like myself. We undertake our research at the University of South Australia. There's a picture of the beautiful facilities we have access to here in Adelaide. We're located in the Bradley building on level 6 and level 8 where our laboratories are. But more importantly is the beautiful team in the photo below the building, which is the team that we work with at the University of South Australia and where we undertake our research. The platform technology that we have at Sementis is protected by a comprehensive family of international patents. And to our knowledge, we are in fact the most advanced viral vector vaccine platform in Australia in development. I have put our website there if you'd like to look a little bit further into the team and our board as well. So the title of this talk was about unlocking Vaccinia as a viral vector vaccine platform. So I just want to take a few steps back and explain that. Now, quite different to what Tim presented, which is a one vaccine sort of single disease focus. We are focused on a vaccine platform technology, and principally a vaccine platform technology is a core technology that can be adapted to a range of different viruses or diseases. And the intent with that is to be able to develop a platform to a certain point and then have a rapid response to an emerging threat, but equally is to provide efficiency in product development and regulatory review. Now, Vaccinia as a viral vector platform, a vaccine vector platform, has long been known for its potential. However, only 2 vaccines have been commercialised using a Vaccinia vector backbone to date, both by Bavarian Nordic. So our previous company founder obviously had a hand in that and knows the technology well. And they have used the modified Vaccinia Ankara MVA as the vector. The 2 vaccines that have been licensed to date have been Jynneos, which are for smallpox and monkeypox, and that fundamentally uses the pox virus backbone to provide a protection against those diseases. The way in which MVA has been used as a viral vector to date is as an Ebola vaccine, which also commercialised by Bavarian Nordic, which is Mvabia. And this is where they've actually inserted the Ebola antigen into the viral vector to use it as a preventative vaccine. So what's interesting about how the company was founded and our technology was that Dr. Howley specifically designed the Sementis Copenhagen vector to address the limitations of MVA. He knew them well, you know, as being part of Bavarian Nordic, and he sought to address them. So the first thing is around the safety of the platform. The first thing was to ensure the safety, that the vector was made replication deficient via a targeted genetic deletion, D13L. And the intent behind this was to obviously make the vector safe, but retain the highly immunogenic characteristics of the vaccinia virus. And this is very different because MVA uses an attenuation, a passaging to make the vector safe. But in doing that, you know, there's always the risk of losing the immunogenic properties of the vector. The second thing is that the SCV technology that we work with is manufactured in a proprietary, well-characterized continuous cell line, Chinese hamster ovary cells, or CHO, as it's known. And really the intention there was to enable the vector to be grown to high cell densities, to have large-scale batches suitable for population scale deployment, enable the production using established platform technology, equipment infrastructure, allowing rapid scale-up, and also overcome the need for extensive batch-to-batch testing. And this is different again to the MVA, which is produced in chicken embryo fibroblasts and basically has all the problems that we're trying to solve. So unable to scale easily, expensive to scale easily, and a lot of batch-to-batch inconsistency. So in terms of SCV and the market opportunity that we see for this platform technology, the first thing is that it can be easily adapted to include multiple or complex virus antigens or disease pathogens. And this is really a point of difference versus other viral vectors. Vaccinia has a large genome, and so as such is able to accommodate large genetic inserts or multiple insertions. The other thing is that using this vector, we're able to insert multiple antigens, but we can have a multiple disease vaccine produced in a single manufacturing run. So at the moment, with other traditional vaccine technologies, it's a single pathogen, single vaccine. And therefore, when you want to have different pathogens or different viral elements included, you need to make those single vaccines and combine them. And that's a real problem when it comes to formulation and the impact on the immunogenicity of those individual components. So this is a real benefit with our technology that we're developing. The other thing is that, as I said before, we've used a genetic deletion to render the virus replication deficient versus attenuation. This precise gene deletion does not affect the antigen expression versus attenuation by serial passaging as used as MVA. But also today, with an extensive preclinical body of work we've done, which I'll show you in a moment, we've demonstrated a strong and sustained humoral and cellular T-cell response. And this really is important in terms of reducing the need for perhaps ongoing boosters that we're seeing with some of the newer technologies, but also the importance of having a strong T-cell response to be able to utilise this technology for diseases such as cancer. The other component that is really a key value proposition for this technology is the scalable and cost-effective manufacturing process. CHO is currently used for 80% of the global biological protein production, so it's well established, it's well characterised by regulators. You know, it's something that people are familiar with. The equipment is known and able to be easily scaled. And this is really important in terms of being able to scale up to population demand. So with an event like we saw with COVID, actually being able to manufacture enough product. And this is one of the challenges at the moment with the current MVA monkeypox vaccine. They cannot produce enough. But also importantly is that it can be cost effectively scaled, and we think that's critical to being able to enable access to lower middle income countries for vaccines using our platform. So just to give a bit of a sense of where we see the opportunities, the types of opportunities for our platform technology, it's clear that we could offer an alternate pox vaccine for the global stockpiles in the ongoing global response to pox-based viruses such as monkeypox. We are really confident that we have an Australian-owned viral vector platform for pandemic preparedness and response, and really having a sovereign Australian capability that we could access in future. We obviously see the opportunity for our platform technology in infectious disease prophylaxis, particularly for more complex antigens or disease pathogens where we need to house multiple or more complex antigens. And again, finally, as I mentioned earlier, because of the efficient T-cell responses and durable T-cell responses, we see the opportunity for use of our technology for treatment for chronic diseases such as allergy or cancer. Just to give you a bit of a feeling for where we are in terms of the development of vaccines or utilisation of our platform and the pipeline we actually have now developed an extensive preclinical data set supporting our SCV platform technology. Along the top, you can see the sort of progress we've made along the development path for different vaccine candidates. We've got several candidates in development. We've had a Zika chikungunya vaccine combination. We had a COVID-19 vaccine that we've been developing and evolved to a multi-component COVID-19 vaccine to leverage the unique characteristics of our platform, which has been funded by the MRFF. We have a smallpox, monkeypox candidate. We're very fortunate in that we've been in a technical evaluation with a multinational company for several years now, and they've also evaluated a technology against 2 different diseases, which remains confidential, of course, to the company. And we've also been developing a peanut allergy therapeutic treatment as well. The main challenge for us as a company has been that we are limited in moving to toxicology and phase 1 clinical studies because it's the manufacturing process development, as Tim alluded to in his presentation. Manufacturing of biologicals is complex, and so that's the step we're currently focused on. But I'm really confident with the work we've been doing over the last 12 months that we have found a feasible pathway to manufacturing, and the next step is to finalise that manufacturing pathway to enable production of phase 1 material and first in human phase 1 clinical trials in the next 2 years. So just to close out this brief whistle-stop tour of Sementis and our technology, I just wanted to share with you some of the collaborations and the capabilities and skill sets that we've developed and we've accessed both locally and interstate. And, just to give people a sense of what you could perhaps come to us and discuss with us if you're interested, or if we can help you in any way in helping you to develop your vaccine or your technology. So Sementis ourselves, myself and Anne particularly, we have a long history in product development and commercialisation, and I guess particularly around project planning and management, a critical skill set in any product development. We, through Anne, have extensive biomanufacturing experience and regulatory strategy experience between the two of us. Also, in terms of implementing quality management systems which are fit for purpose for a biotech or a research group starting out, it's really important to have your information stored in the correct way and documented in the correct way, because fundamentally that is the core value in your company at an early stage, your intellectual property and know-how, and recording and capturing that is absolutely critical. So we've got a lot of experience in that. And of course, between us, a lot of experience in multinational pharmaceutical and vaccine development companies, particularly managing collaborations and how to position technology. With our team at the UniSA and the wonderful group of researchers we work with, in particular, the experience in that team that we've been accessing and developing is our molecular biology and genetic engineering expertise, particularly around vaccine construction and validation. Preclinical evaluation in mice has become a specialty, absolutely. We've also done a lot of cell-based biological assay development, vaccine and virus testing. And as I mentioned before, we've done an awful lot of biomanufacturing process development in-house. We have worked as part of an international tripartite collaboration over the last 12 months to further develop our process. We're unable to access the skills locally, but we've done an awful lot in-house and have built some real capability there. I'm also pleased to say we've partnered with Sagency on the next-gen sequencing of our vaccines. And interstate, we have collaborations with ANU, Monash and CSIRO. For ANU, we've been working with them on rapid construction techniques, rapid vaccine construction techniques. With Monash, a wonderful group doing virus imaging with Dr. Faseli Koulibaly, and at ANU, Professor David Sharkey. And we also have an excellent research collaboration with CSIRO, where we've been also doing manufacturing process development with them. So, look, that's just a whistle-stop tour of Sementis, our technology and our collaborations and skill sets. We've done a lot of international collaboration beyond that with the NIH, as I say, with CDMOs, but also with these international commercial partners as part of a technical evaluation. And so, as part of growing our ecosystem, both locally and nationally, myself and the team are very happy to chat to anyone if we can be of any help. So with that, I'll hand back to Yvette.
Thank you. Yeah, this time my video works. not zooming in and out. Thank you for that, Leanne. And one of the questions I had is around if you could share some of your key partners here in South Australia and globally you've been collaborating with on critical skill and expertise, but that was a very elaborate answer you just gave us. So I'll leave that. Another question, coming here back to South Australia, in 2019, I think, taking on the role as CEO, what are the key challenges you faced? And could you share a little bit more to this group, how you overcome them?
Look, I think one of the challenges that we all face in biotech companies in Australia is the funding environment. I think as you make a transition from research or from an academic sort of, if you're a spin out to a biotech company, funding is a bit of a challenge. So I think I'm really encouraged with the federal government and some of the changes and evolutions they've made in funding programs in the last 12 months. So a great example was MRFF. We actually applied for that as partnership with the University of South Australia, but they've now opened up that program that you could apply as a company. And that's so important, I think, in being able to enable that translational work you need to do and really enable that move that you need to make from research to development, which is quite a different, sort of skill set and a different focus, as you make that move. I think also from a private investment ecosystem in Australia, it's improving and it's growing, but it's still quite challenging compared to, the sort of funding quantums and the risk profile of investors in Australia. So we've still got a little way to go on that. But again, I think there's some great evolution on that in some of the investment groups sort of growing in Australia, and also some of the government programs that are really getting behind investment in startups, but also this really commercialization. So I think that's been one of the key challenges. I think the other thing is for any company is making that transition from research to development. It's quite research is very much an exploration, a sort of, you might go in different directions to answer certain questions. When you're in the development phase, it's a very focused body of research. And you need a real project management sort of, quite a disciplined approach to it. And also because I think as a company, you've got you accountable to your investors. And so they need to see progress against milestones. So I think making that transition is challenging, but I'm actually really proud of our team. We have worked really hard over the last 12 months to make that transition. And I alluded to documentation quality management systems, the way you capture your data and your IP is so important in establishing the foundations of a company. So I think they're probably two of many challenges. I think the other thing is frankly, as much as I'd love to say, and I've come back to South Australia, I'm Australian, I want Australian companies and technologies to be successful. I think the recognition that we don't always have all of the skills and expertise we need in Australia is a challenge, and I always feel a little bit disappointed. So I was incredibly disappointed to have to go overseas to do some of our manufacturing process development. I'm desperately trying to bring that home, but I think it's just a reality of working in a global field like we do in vaccine development. So that's been a challenge, and but I think we're with the presentation from Tim today, there's other groups in Adelaide, and other groups nationally that are building great vaccine capabilities. So I hope we can sort of band together and leverage that a bit more.
And when you were talking about those new sources of funding opportunity with tapping in, like the MRFF and some other examples, what would you recommend other companies that are seeking new funding, how to find them, how to seek support, or what the challenges were you faced to finding and being successful in them?
I think there's two bits. I think there's funding and there's investors, and I think they're quite different approaches and strategies. So look, I think in the grant funding environment, I think you really need to be really clear on how much money you need over what time horizon, and you need to be bold and go out and find that money. And I think, I just want to call out the State Department of Trade and Industry as they were. I think their names changed in the latest election, but there are groups like that, partnering with groups like that, with MTP Connect. We've had amazing support from MTP Connect who, you know, and both of those groups have provided amazing support to us in developing our grant applications. Also, I want to acknowledge the Chief Scientist of South Australia, Carolyn McMillan, and her team. They have been an amazing sounding board. And I think all you can do sometimes is, on a grant basis, is really be thoughtful about the grants you're going for. Go for the right amounts, because you can spend a lot of time writing little bits and pieces of grants. So choose the meaty grants, but really think about how you build your stakeholder network to support you. I'm not saying we've been successful with everything. We've really been, you know, I'm proud of this, the grant money we've brought in, but I think I really, you know, have appreciated the support of our stakeholder groups, both in South Australia and nationally, to improve the quality of what we're doing and to support us in going for those grants. I mean, on the investor side, I think, you know, it's really, you need to get a great pitch deck together, and you need to sort of find some friendly external investors who aren't necessarily the ones you're going to for investment, but will help coach you along the way and give you honest feedback. The number one thing that I look for is people just giving me unvarnished views of our technology and our company. That's the only way we can get better. I'm not scared of it. I embrace it, and I would encourage everyone else to do that too. Listen, get the feedback, get the insights, make yourself better, whether it's from your stakeholders on your grant or from a friendly investor who will tell you honestly what they think about what you're doing.
Thank you, thank you. I know you're still mentoring and advising other startups, but thank you today, Leanne and Tim, for presenting today, sharing the work you're doing and the insights from that. Also, thank you everybody who joined us today here or will be joining us virtually. So this is recorded, the webinar, and we will put it on our website, LA Biomed City webinars tab, so you can always go back or share it with relevant partners or researchers or clinicians in the future. Thanks again, and I wish everybody a lovely evening.